Hello everyone. This is the last lecture of module 9, and in this lecture I will be discussing about antibacterial and antiviral drugs. So particularly I will focus on some major properties of antimicrobial drugs, followed by development of antibiotics and their mode of action, and also I will be discussing antiviral drugs, and at the end how bacteria, particularly how they develop resistance against different types of antibiotics. So let us discuss together first about the general characteristics of antimicrobial drugs. So there should be some important properties of an antimicrobial drugs. So first of all, there should be selective toxicity, because when we are trying to target microbes, for example, in this lecture mostly we will focus on bacteria as well as virus. So we are targeting them when they are present inside our body, that means when we are getting infected. So as a result of that, we have to develop some kind of antimicrobial agent, right? So antimicrobial agent, so that they will not create any problem to host cell. Okay, so they should be safe for host cell, or I would say host physiology, but they should be specifically it should target microbes. So that should be a very important property of antimicrobial drug. Then only we can utilize. Just if I give you one example, like bacteria, when they infect us, so then we can try to target some of the some particular activity, some particular metabolism inside bacteria. So for example, we know that the ribosome, they differ, eukaryotic ribosome and the prokaryotic ribosome, they differ. So as a result of that, if we can develop some kind of antimicrobial agent that can specifically target 70S ribosome, like 70S ribosome. So this is present in bacteria. So 70S ribosome is made up of the 30S and the 50S subunit, like small and large subunit. But in case of eukaryotic ribosome, we have 80S ribosome in our cell. It's a little bit different. So as a result of that, if we can target specifically this either 30S or 50S subunit, that would be very beneficial, and we can use that kind of agent for controlling bacteria. Then the next one is the therapeutic dose versus toxic dose. So we have to be very careful about the dose also, because it should be functional, it should work at very low concentration, because as I just mentioned that there are some difference between eukaryotic and prokaryotic ribosome, that means our ribosome and bacterial ribosome. But if you remember, like we discussed, the fundamental process of protein synthesis is same. So as a result of that, there are a lot of similarities as well between our ribosome and the bacterial ribosome. So as a result of that, if you use a very high concentration of this drug, it might target our ribosome also. I'm just giving one example, taking a ribosome into consideration, but it is true for many other cases. So as a result of that, we should try that some microbial agent that should work against microbes at very low concentration, that is the therapeutic dose, right? The toxic dose, that means it's very high concentration, it can create problem with the host as well. Then another very important point is narrow spectrum versus broad spectrum drugs. Because what happened, for example, if someone is suffering from some bacterial infection, all the time it is not possible for us, or I would say for the medical person, it's not directly recognize a specific bacteria. What is the specific bacteria which is causing the problem? Like from symptoms you can guess, but without proper test it is not always possible to understand the exact genus or species of that particular bacteria. So as a result of that, there are different types of microbial agent, narrow spectrum and broad spectrum. Broad spectrum will work against different types of bacteria. For example, I would say gram-negative bacteria, gram-positive bacteria. Like I would say some antibiotics, if I say ampicillin, this is kind of type of penicillin. So ampicillin is more broad spectrum compared to penicillin. Penicillin is the natural one, first isolated and first discovered antibiotic, but the ampicillin is little bit derivative of that penicillin, but it is broad spectrum comparison to penicillin. Now the next one is cidal and static. That means I would say this is sometimes say bactericidal and bacteriostatic. So as a result of the cidal means it will kill pathogen. So for example, kill bacteria. And static means it will inhibit growth of pathogen. Now the thing is, sometime this is I should mention that some of this cidal microbial drug, antimicrobial drug, if we use at very low concentration, it can also act as static or bacteriostatic, for example, against bacteria if we use, right? So some of the antibiotics we use, they are bactericidal, and few of them they are also bacteriostatic. And now this is just some general features about antimicrobial drugs. Now I will be discussing about the development of antibiotic, or first the discovery of antibiotic. So here, like as many of you know, that Alexander Fleming in 1928, just I am mentioning that year so that little bit idea you will get that at what time point this antibiotic developed. So 1928 discovered penicillin. This is very important, I would say, in terms of I would say revolution in the field of medicine, because this is a powerful drug like any antibiotic, but this is the first antibiotic discovered by Alexander Fleming. What he observed, like he was working with some bacteria, and he found, so as you can see, this is some kind of, this is called petri dish, petri plate or petri dish. Now here if you streak some bacterial culture, that means you are just putting some bacteria with some kind of sterile material. You are just putting this bacteria here, and as you can see, something like this way. And then over the time, this bacteria will grow, and I would say after few hours, maybe after one day, you will be able to see some of those bacteria. As you can see, this whitish substance, this is nothing but bacteria. But what Alexander Fleming noticed, that in one of his plate, that some mold, this is mold, that means some kind of fungus. Fungus actually grow inside the plate, and more interestingly, he pointed out, he noticed that if you see just surrounding the mold here in this position, for example, in this position, no bacteria, right? No bacterial growth observed just beside this mold. So that means some bactericidal agent, something is coming out from this mold and which is preventing the bacterial growth. This is kind of great discovery. And in this way, penicillin was discovered. Later on, it Alexander Fleming and other scientists, they established together that this is penicillin secreted from Penicillium mold. This is Penicillium notatum, right? Penicillium mold, and it prevents the growth, or I would say it kills the Staphylococcus aureus strict nearby. So this bacteria, they are working. This is S. aureus. So this is some gram-positive bacteria, S. aureus. So this is just the starting of penicillin discovery. But the thing is, if you see, I just mentioned that penicillin was secreted by Penicillium notatum. But Alexander Fleming, he didn't purify this antibiotic. Rather, it took a little bit of more time, and later on, I would say a few years later, Howard Florey and Ernst Chen. So in 1939, they purified penicillin from Penicillium notatum. It's a fungus, okay? And penicillin is like some microbial agent which is preventing the growth of bacteria, here particularly Staphylococcus aureus. So this is, I would say, this is antibiotic, preventing bacterial growth, so antibiotic. And not only that, what they have done, so they injected this penicillin, injected to mice infected with streptococci. This is again another group of bacteria, streptococci and staphylococci. So they did it separately definitely, but they injected this antibiotic penicillin, right? Injected penicillin to mice infected with Staphylococcus as well as Streptococcus bacteria. What they found that, so this is very important. So that means purified antibiotic, when it is injecting, it is protecting host, that means in this case mouse, from the bacterial infection. And later on, human trial also happened, and finally I should mention here in 1945, like all of them, right, Alexander Fleming, Howard Florey and Ernst Chen, they received Nobel Prize for their discovery of penicillin. And as I already mentioned, it is a like real milestone in terms of treatment, in terms of from medical point of view. And it inspired other scientists also to isolate different types of antimicrobial agents from microbes, particularly from fungus. And later on, what happened is another scientist, Swayam Prabha, so he purified, so he also purified another important antibiotics are called streptomycin, purified streptomycin from one fungus called Streptomyces griseus. So this is a fungus. So Swayam Prabha also received Nobel Prize because of this work, Nobel Prize in 1952. So as you can understand that This is really important discovery for the human society. And streptomycin also act against different groups of bacteria. And here it is just starting point, and it continue, and finally there are many other antibiotics like chloramphenicol, neomycin, all are isolated from different types of microorganisms, right? And now I will be focusing on how this antibiotic work, their mode of action, okay? So particularly like cell wall synthesis inhibitor. So when I was talking about the specificity, like as you know, in our body, in mammalian cell, like in human cell, we don't have cell wall. We have just plasma membrane, cell membrane. So as a result of that, if some drugs which can specifically target cell wall, so it is very good for us. So we don't have cell wall. So first antibiotic discovered by Alexander Fleming, that is penicillin, particularly if you say penicillin G, this is the natural penicillin. So this antibiotic acts against peptidoglycan. So as you know by now, that peptidoglycan is a major component of bacterial cell wall. Even plant cell wall, they don't have peptidoglycan. So now if we see that this penicillin G, it has high activity against most gram-positive bacteria. As you know, in gram-positive bacteria, we have more peptidoglycan layer also, and little bit low activity against gram-negative bacteria. So this is the penicillin structure, as you can see, and this part is very important. This is the beta-lactam ring. This is very important. I will discuss in more details. This is called beta-lactam ring of penicillin. Now, what happened? All are good. Like this is working against this peptidoglycan synthesis. But some bacteria, they have some machinery, some enzyme that can break this beta-lactam ring. So as a result of that, those bacteria, they are resistant against penicillin. So this enzyme is called penicillinase. That means it breaks penicillin. That's why penicillinase. Or it is also known as beta-lactamase. So this is some enzyme which can break this, this antibiotic. But now I should little bit discuss in more details that if you can remember a little bit about the peptidoglycan structure, that we have this N-acetyl muramic acid and N-acetyl glucosamine, N-acetyl muramic acid, N-acetyl glucosamine is repeating subunit of sugar. And then another chain of the same repeating polymer, something like that. And now if you see from the N-acetyl muramic acid, short polypeptide chain is coming. Although this amino acid is little bit different from our natural amino acid, some of them are D form, like D amino acid. And similarly from here also amino acids are attached with this N-acetyl muramic acid. And you can see some peptide linkage, like cross-linking here. So as you can see, I mentioned before that glycine 5 or some direct cross-linking present in between this short polypeptide chain, and that finally form this mesh-like structure. So similarly from here also amino acid is coming, right? Amino acid is coming this way from this NAM, and from here also coming like this, and again this kind of cross-linking happening. But this penicillin, it prevents the synthesis of this, like this step, it stops. So as a result of that, peptidoglycan cannot be synthesized, and that's why this is very powerful drug, right? Now if I mention little bit about different derivatives of penicillin. Penicillin G is the natural one. For example, ampicillin. Ampicillin is active against both gram-positive and gram-negative bacteria, and it is also acid stable. We have to be careful about this also, because see, most of the antibiotics we take orally. So as a result of that, if antibiotics they are acid stable, that is advantageous for us. So in stomach we have very low pH, very acidic condition. So as a result of that, as you can see, that penicillin G is destroyed by acid. So it is very difficult through taking penicillin G through orally. But ampicillin we can take it orally. And on top of that, another modification happened in this. This is also penicillin group of antibiotics. This is called methicillin. So methicillin is, very interestingly, this is penicillinase resistant. That means the beta-lactam is resistant, but less active than penicillin G. And it is also acid labile. That means it is not resistant to acid. But one important feature here is, it is beta-lactamase resistant. As I mentioned, that this enzyme can break down beta-lactam ring. But unfortunately, when methicillin was used for a long time, again some bacteria, they develop resistance against methicillin also. So as a result of that, you can understand bacteria, they are also clever. They have many different types of strategy to combat this antibiotic treatment. I will be discussing those at the end of this lecture. Now, I should concentrate on protein synthesis inhibitor. I just mentioned about cell wall synthesis inhibitor, or particularly peptidoglycan synthesis inhibitor. But if you see, there are many antibiotics which target bacterial ribosome, and that is also very useful. So as you can see, tetracycline, for example, like there are different forms like chlorotetracycline, doxycycline. This is I would say derivative of tetracycline. So tetracycline, it work against bacterial ribosome. Particularly, I would say its work against 30S ribosome, 30S ribosome. So as a result of that, during protein synthesis, as you know, the 30S and 50S, they bind together along with the mRNA, and therefore translation start. So but if it bind to 30S ribosome and it prevent protein synthesis, so as a result of that, this antibiotic specifically will inhibit bacterial growth, because they cannot synthesize their own protein, right? So this is tetracycline, and here as you can see, some little bit of modification here, like for example, this OH group and this chlorine group, this is not present in tetracycline. Tetracycline lack both of these group, but in doxycycline, this is it's now also we are using this antibiotic. So doxycycline, it consists of tetracycline with an extra hydroxyl group here, and on the other hand, chlorotetracycline, they have extra chlorine group here, but their mode of action is same. So now in this group, another antibiotic I can name, that is streptomycin. This is another antibiotic that act against 30S ribosome. Their target is 30S ribosome again. And now not only 30S ribosome, there are other antibiotics. For example, I would say erythromycin and tetracycline already I mentioned, that is not the right one. So erythromycin and what is that? Chloramphenicol, right? Chloramphenicol. So and chloramphenicol. So both of these antibiotics, they target 50S ribosome of bacteria, 50s ribosome. So specifically, they create problem with 23S rRNA present in 50s 23S rRNA present in 50s ribosome, right? So that's why they are powerful antibiotics, and there are many antibiotics available in in shop, like in medicine shop. They are actually targeting bacterial ribosome. Now, those two are the, I would say, major class of antibiotics. Like one of those group, they are inhibiting peptidoglycan synthesis, and the other group, they are inhibiting protein synthesis in bacteria. And some other antibiotics are also available. For example, inhibitors of nucleic acid synthesis. As you can see that norfloxacin and ciprofloxacin. So here this is the norfloxacin. So they inhibit some enzymes. Particularly, I would say they inhibit topoisomerase enzyme, right? And topoisomerase is important for bacterial replication. So as a result of that, if they inhibit topoisomerase, then again bacteria cannot replicate themselves. So this is like one way how this antibiotic work. And some other group of antibiotics, they are called antimetabolites. So for example, trimethoprim, dapsone, and isoniazid. So they are common example of antimetabolites, and I should also mention most of them are bacteriostatic, right? They just prevent the growth of bacteria. And particularly, I would focus on isoniazid. So this is isoniazid structure. It inhibits mycolic acid synthesis. So what is mycolic acid? So as you know, Mycobacterium tuberculosis, so it causes TB or tuberculosis disease to human, right? But this bacteria is little bit different from classical gram-positive bacteria as well as from classical gram-negative bacteria. So they have some their special features. One of the special feature is they have mycolic acid on their surface. But this antimetabolized, this agent actually inhibit the mycolic acid synthesis, and that's why this drug isoniazid, so it used against tuberculosis. This is very important drug against tuberculosis. Okay. Now, rifamycin and rifaximin, they also inhibit, as you can see, DNA-dependent RNA polymerase. So this is bacterial DNA-dependent RNA polymerase, okay? Because we have also human RNA polymerase, but this drug shouldn't impair the function of human RNA polymerase. This is bacterial DNA-dependent RNA polymerase. Now, before discussing these antiviral drugs, I should mention, as you can see, this antibiotics, they are targeting different steps of bacterial, bacterial growth, for example, protein synthesis or cell wall synthesis. But I mentioned, in some other lectures that we have huge amount of beneficial bacteria in our body. Like in our body, huge amount of bacteria present, which are actually helping us by different method, right? So as a result of that, unnecessary, we shouldn't take antibiotics. Only if it is absolutely required, if doctor prescribed antibiotics, then only we should take. Because these antibiotics, when they are acting against peptidoglycan synthesis or against protein synthesis, but they are also killing those good bacteria, those beneficial bacteria present in our body. So that is one problem also. So that's why very judiciously we have to take antibiotics, not randomly, like sometime without anything we shouldn't take antibiotics. It will be more problematic than the than better result. And now antiviral drugs. This is little different from antibiotic, because bacteria, they have their own machinery, their own system, and we are targeting them. For example, peptidoglycan, for example, their ribosome. But as you know, virus, they have very little thing. They have little bit of their own genome, either DNA or RNA, and some specific protein present on the surface. But mostly they are utilizing host machinery, and that is the problem to target virus compared to bacteria, right? So this is kind of little bit challenging for us, to target virus and to combat with virus. So let's see how we can do that. So most antiviral drugs disrupt critical stages in a virus multiplication cycle, and probably the most publicized antiviral agent is Tamiflu, and this is the chemical component, the main thing present in Tamiflu. So now what Tamiflu does, right? So this is antiviral, many of us we know. The Tamiflu inhibits the viral molecule called neuraminidase, and this is which is essential for release of newly synthesized influenza virus particle from the host cell. As you know, I discussed during the virus lecture, virus, they are hijacking our system. They enter inside our cell, host cell, and they are making their own parts, and finally they will generate virus particle, and they should leave the host cell and infect again nearby cell or the surrounding cells. But neuraminidase, they are preventing the release of newly synthesized influenza virus particle from the host cell. So if you stop the release of this virus, newly generated virus from the host cell, so they cannot infect the surrounding cells. So that way we can also prevent viral growth, okay? And that's why Tamiflu received much attention during the 2009-10. During that time we had H1N1 influenza pandemic, okay? So that's why this is very famous, like Tamiflu. And there are many other antiviral agents also available in market now. But now I will mostly focus on mode of action of different anti-HIV agent. There are lot of progress happen with HIV disease, right, and how to prevent that. Now there are many good remedy available against human immunodeficiency virus. So as you can see, this is HIV virus. So now there are many important steps we have to target. I am just taking HIV as an example, but this is true for many other viruses as well. But we have to develop virus-specific drug also. So now infection begins with HIV fusion. So as a result of that, we can use some fusion inhibitors so that the virus will not be fused with the host cell itself. And so that is kind of one important drug, right, that can work against HIV. And the next step is, once inside the host cell, HIV uncoats and its reverse transcriptase forces the host to make DNA from the viral RNA. As you know that HIV, it has RNA as its genetic material, and it encodes, the virus itself encodes the reverse transcriptase enzyme. So as a result of that, first it will make RNA to DNA by reverse transcriptase, okay? So this step is called reverse transcription. So now there are lot of progress with RT inhibitors. That means the reverse transcriptase inhibitors that block this step. So this is very useful drug, I would say. And one of the most important antiviral anti-HIV agent is azidothymidine or AZD. So as you can see, it has very similar. This is, I would say, nucleoside analog, okay? Nucleoside analog. As you can see, some sugar is there, thymidine group is also there. And only thing is, very carefully notice here, the 3' here it should be OH in order to extend the chain during the polymerization reaction. Instead of that, it has azide group here instead of OH. So therefore, it will stop the synthesis of the chain. So this is just there is some kind of similarity when you will be learning about the dideoxy chain termination discovered by discovered by Sanger for DNA sequencing, right? So, but here because of the azide group, it will not be extended by enzyme polymerase. Another important thing I should mention, this is prodrug. Prodrug means, so prodrug, this is nucleoside analog, but for polymerization or for DNA, RNA, what is the monomeric unit? That is nucleotide. But nucleoside analog we are using here as prodrug because the permeability is better, so that this drug can go inside the cell in much efficient way. So prodrug, so permeability is better. But after entering inside the cell, then the phosphate group will be added. Then it will be behaving like nucleotide, and therefore it will work against these viruses. But now the question is, if you carefully listen, the question will come to your mind that whether it will impair the polymerization by our human DNA polymerase, because then that will inhibit the synthesis of human DNA also, right? But I would say, yes, theoretically it is possible, but the thing is, human DNA polymerase, it is more powerful. It has proofreading activity. Whenever it will check that some wrong nucleotides are there, so it can remove it and properly it can adjust accordingly. But compared to that, I would say reverse transcriptase, it's not that much powerful in compared to human DNA polymerase to take care of this thing. So as a result of that, it will stop the function of reverse transcriptase specifically. And then some protease inhibitor also can be used, because the viral DNA is transcribed and translated into linear polypeptide, and that is cut to release different viral protein so that virus particle can be assembled. But now if we can use some protease inhibitor specifically, so that this viral protein will not be processed and it can also be used as drug. All those things are actually being used nowadays. And the last one here is the viral DNA, particularly for HIV, is added to the host DNA by some enzyme called integrase. So if we can develop integrase inhibitor, it can also be powerful against anti-HIV, not anti-HIV, it's powerful against HIV. Okay. So these are the different approach to target human immunodeficiency virus. And this is the last slide. Here I would like to discuss about the antibiotic resistance, which is a very, which is a real problem now. So as you can see, this is bacteria, right? And now if you see, we are targeting some critical steps inside bacteria. For example, if you are targeting a critical enzyme, this is the critical enzyme here, as you can see in bacteria, right? For example, topoisomerase, as I mentioned. Now, when you are applying antibiotic, it is going inside the bacteria and then it binds to the critical enzyme and it inhibits the function of critical enzyme, and therefore bacteria cannot survive, it cannot grow. This is the mode of action. But bacteria is also very clever. What it can do, so some resistant bacteria, I would say resistant bacteria, so they can increase the expression or the production of this target molecule. So if they can increase the amount of target molecule, particularly here the critical enzyme, a lot, then how much antibiotic we will be giving, like that particular dose, very high dose can be a problem for the host cell also, right? So this is one strategy bacteria they use and they become antibiotic resistance. And there are many other, other method by which they can be antibiotic resistance. For example, they can alter the critical enzyme itself. For example, because of some mutation, now your antibiotic is not acting against the critical enzyme, your target, right? So for example, mutation in the particular gene that produces target protein. or enzyme. Okay. And then protein that degrades drug. This is also very important. Some bacteria, they are resistant because they have some protein which is degrading the antibiotic. I already mentioned that beta lactamase. So beta lactamase, they break beta lactam ring present in penicillin group of antibiotic. So as a result of that, those bacteria now they are resistant against penicillin. Now I would particularly mention some strategy, not just one thing you have to use. Now, like human, we are also dealing this problem in a different way. For example, now one antibiotic, you might know this name, augmentin. Generally, if someone is suffering a lot, then augmentin can be used. Doctor might prescribe this augmentin. So augmentin is just mixture of penicillin plus clavulanic acid. So this is amoxicillin. So amoxicillin is also a derivative of penicillin. So amoxicillin and clavulanic acid. So what it does, the amoxicillin or penicillin, it prevents the cell wall synthesis, but the problem is some enzyme beta lactamase is present in some resistant bacteria. That's why they are destroying this penicillin or amoxicillin, whatever you say. But the thing is, this clavulanic acid, which is present in addition to amoxicillin in this drug augmentin, this clavulanic acid will destroy the beta lactamase. It will break down beta lactamase. So as a result of that, now that particular bacteria is not resistant to the antibiotic itself. So I hope that you understand that in augmentin we have both the penicillin as well as the beta lactamase inhibitor, and that's why this is very powerful antibiotic. And another strategy very often used by bacteria, that is, we are trying to treat by antibiotic reagent, right? Antibiotic drug. So it should go inside the bacteria and then they should stop critical step inside the bacteria. Now bacteria will try to survive. So what they will do, so this is efflux pump, as you can see, this is efflux pump. So they will increase the amount of efflux pump on the surface of these cells in the bacteria itself. So therefore, like we are trying to give more and more antibiotic and it is going inside the bacteria, and bacteria is removing those antibiotic from inside to outside. So therefore, this is another very important strategy of bacteria, right? So that's why this antibiotic resistance is a big problem, right? So properly antibiotics should be used, because misuse of antibiotics can lead to more antibiotic resistance, because in the last lecture I mentioned that bacteria, they can transfer their genetic material in a horizontal gene transfer manner. So as a result of that, some antibiotic resistance gene can be moved from one bacteria to other bacteria, and that is increasing bacterial resistance, right? So this is all about antibiotics and antivirals and the resistance against antibiotics. That's all. Thank you very much.